Swayam Prabha. Digital India. Educated India.
Hello everyone, welcome to the next lecture in the first module of Plant Biotechnology course. In this module, we are talking about basics of plant tissue culture, which would include variety of topics like various biological and chemical or the physical factors that impact the plant tissue cultures, totipotency, differentiation, somatic embryogenesis, micropropagation, and so on. My name is Manoj Sharma and I am working as an assistant professor of plant biology at Jawaharlal Nehru University. In JNU, I teach plant biotechnology course and genetic engineering applications at School of Biotechnology. These lectures have been reviewed by Professor Kashmir Singh from Department of Biotechnology, Punjab University, Chandigarh, and this project has been sponsored by DTH Swayam Prabha, MHRD, New Delhi. The growth and morphogenetic responses in vitro cultures depends largely upon three factors. One is the physiological status of the biological material that is used to initiate the in vitro cultures. None is the chemical components or the nutritional composition of the culture media used, and finally the physical components or the environment that influences the tissue culture responses. Now, in the last lecture, we talked about the health of the explant as well as the health or the genotype of the donor plants, and several other parameters we discussed related to the biological materials. Today, we will talk about the composition of the plant tissue culture media or the nutrient composition. And finally, by the end of the presentation, we will talk about the physical component that influences the tissue culture responses. Now, selection of right composition of culture media is very important for establishing plant tissue cultures. And there isn't any one particular optimized media composition that would work for all the plant species. Different plant species or different group of plants have their own specific requirements, specific growth requirements, and therefore optimization of species specific media composition is crucial. There are several different nutrients or chemical or the salts that are used in the tissue culture media, and generally these components can be grouped into the following categories. We have inorganic source of the nutrients, and then we have organic source of nutrients, carbon source, then growth regulators, solidifying agents, and the media pH. Media pH can also be discussed under the several times it is discussed under the environmental or the physical parameters too. The first is the inorganic nutrients. Now, there are several inorganic mineral elements that are essential for normal growth and development in plants. Depending upon the amount of these elements or minerals that are required for optimal growth, these have been classified into two groups, that is macronutrients and micronutrients. The elements that are required in large quantities are categorized as macronutrients, and the other elements which are required in very small quantities or the trace amounts are categorized as micronutrients. So let's talk about first the macronutrients. These elements usually they are required in the concentration which are higher than 0.5 millimolar. In addition to carbon, hydrogen or oxygen, the other elements which fall in this category includes calcium, magnesium, potassium, phosphorus, nitrogen, sulfur. Depending upon the required concentration, and it usually depends upon what is the role of that element in the plant metabolism, like it may be a structural component of the plant body, or it may be involved in diverse functions, and hence it may be required in the higher concentrations. On the other hand, there may be other nutrients which are working as a signaling molecules and may be required at very low concentrations. And the concentration of the same element required for optimal growth may vary among the different group of plants also. So their concentration, the required concentration may vary from one element to another element, and also the required concentration of these elements or the same element may vary among the different group of plants. Now, depending upon the concentration, nitrogen is one which is required in the maximum amount. That is, it is supplied usually in the range of 30 to 60 millimolar. That is the high concentrations in the media. It is usually supplied as nitrates and ammonia salts, and actually it is performing several basic functions. It's a constituents of the nucleic acids, proteins and amino acids, so it is participating in several basic metabolic processes. As per the amount required, the second in the line is the potassium, which is usually supplied in the media in the concentration of 20 to 30 millimolar. Potassium is required for several functions like cell division, stomata regulation, synthesis of proteins and chlorophyll. It also contributes significantly towards regulation of osmotic potential and pH within the cells. Most media contains or it is supplied in the form of the nitrates or chloride salts of the potassium, and the optimum concentration for all other elements is that is the phosphorus, magnesium, sulfur or the calcium. It ranges between 1 to 3 millimolar. Now, calcium is an important constituent of plant cell walls. It is also a signaling molecule and is involved in the regulation of hormonal responses, which play a significant role during the morphogenesis also. Magnesium is a component of chlorophyll molecule and play an important role in the respiration as well as ATP synthesis. Phosphorus is a part of several macromolecules like nucleic acids, phospholipids. It is also an enzyme cofactor, and similarly several other processes is enclosed. It is very much required for the storage and the transfer of energy in the form of ATP molecules, ADP molecules or AMP. Similarly, sulfur is a structural component of the amino acids and enzymes and is involved in the lipid synthesis too. So overall, it is crucial to identify the optimum concentration of each nutrient, which may vary considerably among different species. Next is the inorganic nutrients, micronutrients. So many elements like iron, zinc, copper, manganese, cobalt, boron, or molybdenum, they are required in very minute amounts or the trace amounts. And so usually the concentration which are supplied in the culture media, they are less than 0.5 millimolar, and these elements are categorized as micronutrients. These are usually required in micromolar concentrations. Among the micronutrient, manganese and boron are the ones which are required, whose required concentration is towards the higher end. That is, they are usually supplied in the media around 20 to 100 micromolar, depending. It varies again from the species, one plant species to another plant species. Second in line, depending upon the concentration, is zinc, which ranges from somewhere 5 micromolar to 30 micromolar. And most of the other micro element or the micronutrients, they are supplied in less than 1 micromolar concentrations. In some of the cultures, iodine is supplied at somewhere around approximate 5 micromolar concentrations. Now, iron is usually the most critical among all the micronutrients. It works as a respiratory electron carrier in cytochrome, oxidative enzymes or peroxidases, catalases, and so on, and is critical for the plant growth and development. Similarly, many of the micronutrients like copper, zinc, molybdenum, they work as a cofactor of enzymes or the component of enzymes, and thereby they regulate the metabolism. Many of these micronutrients, they are involved in the DNA metabolic pathways. They are required in the minute concentration, very little concentration in the plants. However, they play a significant role in the plant development, specifically again in the morphogenetic responses. Now, again, as for it was for the as we discussed for the macronutrients, the required concentration of micronutrients, it also varies from one element to another element, and also the concentration of the same element, required concentration for the same element for optimal growth, it varies from one species to another species, or specifically a different group of plants. Inorganic nutrients, whether the micronutrients or the macronutrients, they are usually added in the plant culture media as the salts. However, in aqueous solutions, they dissociate into the ions, that is cations like the magnesium, calcium, potassium, or the anions like the nitrates or the sulfates. So they are supplied, so like nitrogen is supplied in the form of nitrates or the ammonium salts, whereas the potassium is supplied as the chloride or nitrate salts of the potassium, and so on. So that is the two group of the inorganic sources of the nutrients, that is the some of the elements which are required in higher concentrations, they are grouped into microelements, and the ones which are required in very low concentration or trace amounts, they are micronutrients. So the inorganic nutrients in the form of macronutrients or the micronutrients. So the next is organic nutrients, which are supplied in the plant tissue culture media and are required for optimal growth of the cultured cells or tissues or organs. Usually there are two group of organic nutrients, that is vitamins and amino acids. Let's talk about the vitamins first. Now, we know the role of vitamins in our life. Most of the time, when you visit a doctor for the consultation for any condition, we are prescribed vitamins along with the other prescriptions. Now, these vitamins, they work as a coenzymes in various metabolic processes, and hence they play an important role in our body metabolism. Similarly, these are required for growth and development in plants too. However, plants can synthesize most of these vitamins. Now, if the plants can synthesize them, why do we need to supply them in the culture media? Well, intact plants can synthesize them, or intact plants, the complete plants can synthesize all the vitamins. However, cultured plant cells or the tissues, they cannot perform all the metabolic functions of or the processes that occur in a whole plant, and usually they become deficient in some of the vitamins, and hence exogenous supply of the vitamins, it is required along with the media components for growth and survival of the culture tissues. So these are used as a catalyst in various metabolic processes and actually required for the basic metabolism in living cells. Thymin, pyridoxine and nicotinic acid, these are the most commonly used three vitamins in the tissue culture media, and they are used in the concentrations ranging from 0.1 to 0.10 milligram, whereas the nicotinic acid is used up to 5 milligram per liter. So they are required in very small concentrations. Now, Thymin is the one which is absolutely essential and need to be supplied into the culture media. Thymin, as far as its function is concerned, it is an essential cofactor in the carbohydrate metabolism and is involved in the biosynthesis of some amino acids. Whether pyridoxine and nicotinic acids are absolutely essential to be supplied in the media, it is still debatable. However, these are supplied in most of the tissue culture media. Similarly, several other vitamins that are used in many cultures depending upon the species specific requirement, and these includes like ascorbic acid, folic acid, tocopherols, biotin, riboflavin, and so on. And usually they are optimized as per requirement of the species specific manner. Another component is myo-inositol, which is prepared with the vitamin stocks. Now, but myo-inositol as a chemical, it is a carbohydrate or a sugar, but it is prepared with the vitamin stocks. Now, as a carbohydrate, it's a sugar alcohol, and alcoholic carbohydrate or the sugar alcohols, they plants are not able to utilize them or the metabolize them as a carbon source or an energy source. So therefore, the myoinositol doesn't qualify to be a carbohydrate. On the other hand, it is believed to play an important role in cell division because of its breakdown to the ascorbic acid and a pectin, and is absolutely required for growth and development responses in plants. Hence, because of its function, uh, that is usually the responses like morphogenetic responses or other growth and development responses, it is placed with the vitamins. It also participates in biosynthesis of several pectin and hemicelluloses, which are ultimately required for the cell wall biosynthesis. So it play a significant role in the cell wall metabolism also, or cell wall biosynthesis too. So next is the amino acids. Amino acids are not actually an essential requirement in the culture media, as the required amino acids for the optimal growth can be synthesized by the plant cells. However, inclusion of the amino acids or mixture of amino acids in the culture media have been shown to improve the regeneration significantly, at least in some plant species, for which actually it is difficult to establish the tissue culture. Now, amino acids serve as an organic source of reduced nitrogen, which is easily assimilated by the growing cells as compared to the inorganic nitrogen, which is supplied in the form of the salts in the culture medium. Their impact varies among the different species. That means in the cultures of some plant species, addition of amino acid may not make any difference. However, in the others, it can make huge differences to establish and to make the culture successful. These have been shown to make a significant difference in the tissue culture of those plant species which are actually recalcitrant to the culture, or they are difficult to regenerate or do not easily respond to the in vitro culture media. They have significant impact on the cultures of isolated cells of the protoplast in the suspension culture too. Casein hydrolysate is commonly used as a source of amino acid in the tissue culture media. Now, casein is one of the main proteinaceous component of the milk, and it actually accounts for more than 75 to 80% of the total milk protein. It is usually prepared from the digestion or the hydrolysis of the casein protein, which actually supply almost all the amino acids. In addition, it also a source for vitamin, calcium, phosphate, and several other micronutrients. In addition, several amino acids have been tested individually or in combination of one or two amino acids, and have been found to promote the tissue culture, specifically the organogenesis, to a significant impact. Some of these are asparagine, tyrosine, glutamine, arginine, cysteine, etc. They have been used in various culture conditions. Usually, as far as amount is concerned, amino acids are used in up to 100 milligram concentrations, like asparagine and tyrosine specifically. They range up to 100 milligrams per liter. Then we have glutamine and arginine or cysteine. Their range is up to 10 millimolar, 10 milligram per liter. And then we have a glycine, which is usually used in some cultures at the concentration of 2 milligram per liter. So overall, amino acids may or may not make an impact in the tissue culture. It would depend upon the type of species we are working with. Next is the carbon source. So in the beginning of in vitro cultures, most of the cells do not contain or do not have chlorophyll, and their chloroplasts are poorly developed, and hence they lack photosynthetic capacity. This means that these cells would not be able to fix the carbon for themselves. Therefore, in order to meet the higher metabolic activity in the cultured cells or tissues, exogenous supply of the carbon source would be mandatory. Sucrose is one of the most commonly used source of carbon in the plant tissue culture media. It is usually used at the concentration of 2 to 5%. Now, tissue culture media need to be sterilized before these can be used for the actual cultures. Now, for sterilization, either it can be performed by filtering through the micron pore filters, or it can be performed by autoclaving. It has been found that sucrose after autoclaving perform better in the cultures as far as their impact on establishing the tissue culture or organogenesis is concerned. So what is it in the autoclaving that make it better? So actually, autoclaving causes partial hydrolysis of the sucrose into other simple sugars like glucose and fructose. These simple sugars are efficiently utilized by the growing cells, and hence perhaps are the better responses. Well, autoclaving is not the only source for the hydrolysis of this sucrose in the tissue culture. Sucrose is also hydrolyzed by the plant-derived invertase enzymes that are secreted by the growing cells in the media. Another very important factor of the sucrose is to maintain the osmoticum in the culture media. There are several other sugars which are used in the culture media as a carbon source. These are some of the examples. The simple sugars, the monosaccharides like glucose, maltose, galactose, mannose, and so on. These are used as a carbon source. Now, the type of sugar may have a different impact on the type of culture. Like for somatic embryogenesis, maltose produced better result as compared to the sucrose in soybean and alfalfa. Similarly, glucose may promote organogenesis to evoke better responses in some species. Next is the carbohydrates. We have sugars which are actually the alcohol sugars, and plants cannot metabolize sugar alcohols as a carbon source. Therefore, mannitol or the sorbitols, which are the sugar alcohols, cannot be used as a energy source or the carbon source. However, they can be used for the osmoticum purposes only in the media. Earlier, we discussed about the myo-inositol also, which is also a alcohol sugar and a carbohydrate which cannot be used as a energy source in the cultures. However, it plays an important role in the cell wall metabolism and cell division, and hence it has been grouped with the vitamins and prepared with the vitamins. Next is the undefined organic supplements. These are the crude natural plant extracts whose active constituents are not known or are not quantified. There are several natural complexes or undefined organic supplements that have been successfully used in the culture or the tissue culture of plants. These includes coconut milk, yeast, malt extract, banana extract or the potato extract. Then we have orange juice or tomato juice. However, the coconut milk, which is one of the most popular among undefined organic supplements, is quite frequently used, or it was quite frequently used to establish the cultures. And then others several others are also have also been tested in the cultures successfully. Now, use of these supplements was very common during the early years of the tissue culture. However, with the improvement in our understanding about the balance of the inorganic salts, amino acids and growth regulators for optimizing the tissue cultures, use of the coconut milk or any other undefined supplements, it declined. Usually when nothing else worked, then these supplements of undefined makeup can be tested to initiate the tissue culture, to establish the tissue cultures. Now, these are usually called as undefined because their composition is not known. That is, what kind of nutrient does it have is not known, or even in what concentration these nutrients are there, it is not known. Further, even if we know the composition of any particular undefined supplement, its composition may vary with the age of the source tissue, genotype of the source plant from where it is prepared, or even the environment in which the source plants were growing. Hence, one of the major challenge with using the undefined organic supplements in the plant tissue culture is the reproducibility of the results. Therefore, their use is usually avoided if some better alternative of the known compositions are available. So next is the growth regulators. The chemicals that are naturally present in the plant tissues, and instead of the nutritional roles, these chemicals regulate the growth and development in plants, and these are called as the plant hormones or the growth regulators. These are required as a supplement in the tissue culture media to evoke various type of morphogenetic responses or the various types of differentiation in the tissue culture. These are called as the growth hormones. They are required in very minute concentrations ranging from the picomolar amounts to the micromolar amounts. However, their functions or their impact in the regeneration on the in vitro cultures is huge. There are several different hormone categories like the auxins, cytokinin, gibberellin, ethylene or abscisic acid. However, auxin and cytokinin are the most important and the popular ones that are required in the tissue cultures. Others, that the gibberellins or ethylene and abscisic acids, are not something which is mandatory to provide in the tissue culture. However, they may be important to for establishing the culture in some specific plant species. Now, the type of the growth hormone and its quantity varies depending upon the plant species that we are culturing, like tomato plant versus rice plant. Type of the explant also, like if we are culturing starting material is apical meristem or embryo versus leaf, or it is the type of responses that we are looking for, like if we are looking for a de-differentiation to initiate the callousing or to induce the shooting from the callousing or the rooting from the excised shoots. So their concentration and type of hormones it vary depending upon the situation, what exactly we are trying to achieve in the culture conditions. Auxins are integral part of the nutrient media, as they are widely used in the plant tissue cultures for several different kind of responses, ranging from callousings, morphogenesis like shooting, induction of shooting, induction of embryogenesis or rooting, and so on. At the cellular level, auxins have significant effect on the cell division and cell elongation, and at the tissue level they are involved in various developmental processes like apical dominance, abscission, rooting, stem elongation, cell division, and so on. However, in the tissue culture, these are majorly used for the purpose of cell division and differentiation, that may be in the form of organogenic differentiation or the induction of embryogenesis or the somatic embryogenesis. Some of the common auxins that are used in plant tissue culture media are indole 3-acetic acid, IAA, indole 3-butyric acid, IBA, naphthalene acetic acid, NAA, and 2,4-D, 2,4-dichlorophenoxyacetic acid. Now, different auxins usually differ in their physiological activity, and hence they may be required at the different concentrations for the same type of response. Like 2,4-D is more effective for the induction of callus and somatic embryogenesis. IVA and NAA alone are commonly used for rooting responses. However, in combination with the cytokinins, they are used for the shoot proliferation too. In tissue culture, generally at low concentration, they favor, or the use of auxins favor rooting response, whereas at higher concentrations, they induce callousing. The choice and type of the auxins and the amount of the or the concentration of the auxin may depend upon several factors. Like it would depend upon the type of the growth or the development that we are looking for. Like if we are looking for the rooting response versus shooting response or callousing or embryogenesis, then the inactivation of the auxin in the culture media. Now, in media have several different types of the salts, and how auxins are interacting with the media salts, it may some of the components may compromise the activity of the auxins, and then the endogenous level of the auxins in the explant itself, that is the endogenous, the plant tissues have different levels of these hormones depending upon the physiological activity, and that also it also depends upon this endogenous levels, how much we need to give the exogenously. And finally is the auxin sensitivity of these tissues, that how sensitive the plant tissue was. So basically, again, the sensitivity of the plant tissue may vary with the type of the explant, type of the tissue which is used to initiate the tissue cultures or the in vitro cultures. Next is cytokinin. So cytokinin is another group of plant hormones that play significant role in vitro culture establishments. Usually cytokinins are associated with cell division and shoot differentiation, and in vitro cultures they are specifically used to induce the cell division as well as the differentiation of adventitious shoots from the callus or any other explants which are used to initiate the cultures. For these responses, cytokinins are required in low concentration. However, their concentration may vary depending upon what kind of response is required. Kinetin, BAP, benzyl aminopyrin, and zeatin or TDG are some of the common ones which are used in vitro culturing. And this is also a common observation that increased concentration or if the cytokinins are used at a very high concentration, they promote callousing rather than the morphogenetic responses. Now, it's a well-known fact that the auxin and cytokinin interaction regulate the in vitro morphogenesis or responses of the explant or the callus, how they behave when both the auxin and cytokinins are given in the tissue culture. So the general concept is that usually higher the auxin and cytokinin ratio, that is more amount of auxin is used and cytokinins are used at the lower amount, they induces the root regeneration, whereas reverse of this, where that is the increased concentration of cytokinin is used with the lower concentration of auxin, it usually results in the shoot induction or it promotes the shooting response. Though the molecular mechanism of these interactions between the auxin and cytokinins is largely unknown, but the evidences suggest that there is a crosstalk between the auxins and cytokinin during the in vitro organogenesis. Therefore, it is very, very important to carefully select the hormones and optimize their concentrations for effective results. Depending upon the presence and the concentration of other hormone in the media, the use of auxin or the cytokine may change the type of growth or the response it would make. Here is the graphical depiction of the responses of the ratio of auxin versus cytokinin. So here on the x-axis we have auxin concentration, increasing auxin concentration, and on the y-axis we have increasing cytokinin concentration. So higher auxin to cytokinin ratio here in this region, it usually result in the rooting response, whereas higher cytokinin versus auxin ratio, it result in the shooting responses. And in general, if they are used at the equal concentration, it usually results in the callousing responses. Hence, the concentration of any hormone and their combination with other hormones must be tested for each plant species, as each species is unique in itself. However, to some extent, results may be transferred to another culture with careful analysis of the species and how close the species are. But even then, we need to test these results whether it will work in the other species or not. Similarly, other hormones like gibberellins, ethylene, or abscisic acid, they can be used in vitro tissue cultures. Like gibberellins stimulate the stem elongation in natural conditions, and therefore in vitro cultures also they can help in shoot elongation. Ethylene have been reported to impact the somatic embryogenesis both positively and negatively. That means in some species it may promote the somatic embryogenesis, and in other species it may inhibit it. However, these effects are very specific to certain species, and therefore these hormones may not be required in routine tissue culture experiments. Next is the gelling agents. In liquid media or liquid culture media, explants or tissues will simply submerge and will die of anaerobic conditions. Therefore, to take care of this problem, stationary support is mandatory, and the culture media are usually solidified with a suitable gelling agent. Now, a suitable or a good gelling agent should have a few desirable characteristics. Number one, it should be inert. That's, it should not react with any of the media nutrient or media components, whether inorganic or organic. Second is that it should be able to withstand the autoclaving temperatures, as media need to be sterilized, and most of the time it is done by autoclaving. Third is that it should be liquid when it is hot, so that the heat sensitive components could be mixed uniformly and the medium could be dispensed in the desired vessel in the desired quantities. Many of the organic components, maybe the hormones and some other like the amino acids, they may be heat sensitive, and therefore they are usually added when the temperature of the media falls to about 40 to 50 degrees Celsius. Agar and Phyta Gel are the commonly used gelling agents. Agar is a high molecular weight polysaccharide which is commonly made from the red algae, Gelidium and Gracilaria, whereas the gel gum is a bacteria derived polysaccharide. It is usually derived from Pseudomonas eloidea, and it is also commercially marketed with the name of Phyta Gel and or Gelrite. The gel and gum based media is usually clear, transparent, and therefore it makes it easier to observe the changes in the culture. The firmness of the gel would depend upon the concentration of the gelling agent used in the media. Higher is the concentration of the agar or the Phyta Gel, harder will be the media. However, for the plant tissue culture purposes, semi-solid state of the media is suitable. In this state, it do not let the explant sink in the media and also provide enough contact between the media and the growing tissue so that the tissue could extract the or can have the enough nutrients for to maintain its growth and development. So in the media, agar is usually used at a concentration of about 0.8%, and Phyta Gel is usually used at a concentration of 0.1 to 0.2%. Next is the pH of the media. Now, pH of the media is another very important parameter that can have considerable impact on the tissue culture. It directly affects the solubility of the media salts, uptake of the growth regulators, and the gelling efficiency of the agar or the gel and gum is also affected by the media pH. So basically these are the three important parameters which are directly impacted by the change in the pH of the media. Ideally, uptake of the negatively charged ions is favored at the acidic pH, and the positively charged ions is favored at the higher pH conditions. So therefore, even the uptake of the ions by the cultured cells from the media can alter the pH of the media, because the ion exchange is taking place, and change of ionic strength will occur because of this change, because of this exchange. Therefore, though the plant tissue can tolerate the pH in the range of 4 to 7. However, the ideal pH for the most plant tissue culture experiments is 5.8. At this pH, all the inorganic nutrient salts are maintained in buffer form and are available for plants to use. pH directly affect the gelling efficiency of agar or Phyta Gel based media. If the pH of the medium is more than 6, it will result in a very hard solid media where agar is used, and if the pH of the media is less than 5, media will not solidify enough to hold the tissue without sinking. So the impact of the pH is more on agar based media usually, whereas in the Phyta Gel based media, strength of the Phyta Gel remain unaffected at wider range of pH, but certainly there is a effect of the pH that it may result in changes in the gel strength of because of the agar or Phyta gels. Media is usually sterilized by autoclaving, and autoclaving can result in change in pH. Usually it results in fall in pH, making it more acidic. This change also depends upon the carbohydrate source used for the media. If the carbohydrate source used are monosaccharides, like the simple sugars, glucose, maltose, galactose, the change in pH will be more pronounced as compared to when the sucrose, the disaccharide sugar, will be used as a carbon source. Addition of the agar also can increase the pH of the media slightly, and the storage of the media for the longer period of time at room temperature may also result in change in pH. Usually decrease in pH is observed because of the longer storage of the media. Therefore, it is recommended that if we need to store the media for the longer period of time, it should be stored at 4-degree temperature and in dark. Change in pH impact the biological processes occurring in the culture, like initiating cultures, initiating callus cultures, differentiation, morphogenesis, rooting, shooting. So therefore, it is very crucial to maintain the pH of the tissue culture media so that the nutrients are available, gel strength is in a semi-solid state, and all the processes like the biological processes are not affected because of the pH change. Now next is the physical parameters like the influence of light or temperature. So the light is one of the important environmental factor that controls the plant growth, development or morphogenesis in natural as well as in the in vitro tissue cultures. Light is a major component of the culture environment. The three major aspects of light that affect the in vitro cultures are wavelength, flux density, and the duration of the light exposure for which the cultures are exposed directly. It is specifically required for photosynthesis and photomorphogenesis responses. If the callusing is performed in the dark, these are usually transferred to the light for regeneration of the shoot induction or organogenesis. When exposed to light, these dark grown calli slowly develop chlorophyll and start performing photosynthesis. However, the light of high intensity, specifically in the blue light wavelengths, it can have an inhibitory effects on the callus growth or the cell division in some species. And because of this, callus inductions are usually done in the dark. Light requirements varies from one species to another species, or even during the different stages of the plant tissue culture, as we see that for callusing we are incubating them in the dark, and for the morphogenesis or the organogenesis we are transferring them to the light. So light induction is required for the morphogenesis responses. Significant impact of the light on somatic embryogenesis have also been reported in species-specific manner. Like, it is required for the somatic embryogenesis in Solanum. However, in popular species, absolute dark is required to initiate the somatic embryogenesis. Even the quality of the light is very important for the somatic embryogenesis. Like in carrot, while the blue and white light inhibit the production of the somatic embryos, red and green light have a positive impact on the induction of somatic embryos. So in this way, light is also a very important factor which is carefully need to be optimized in a species specific manner. The influence of temperature. So now in nature, plants are adapted to diverse temperature regimes which fluctuate widely during the different seasons or different geographical regions, and therefore the optimum temperature required for in vitro growth, they may vary from one species to another species. Plants are adapted to the large fluctuation in the temperatures during the day and night conditions. Though it is not mandatory to mimic these actual temperature conditions of the day and night in which actually those plants will be growing, however, lowering the night temperature in the culture room help in improving the morphogenetic responses, or more specifically the induction of shooting response. Usually growth rooms are maintained at an average temperature of about 25 degrees Celsius. However, for the plants adapted to the tropical and subtropical regions, the average growth room temperature is higher and is maintained at about 28 degrees Celsius. The tropical and subtropical plants like the rice, cotton or sorghum. Whereas in the temperate plants, it is maintained at a little low temperature, somewhere around 24 to 25 degrees Celsius. The usual day and night temperature in growth room are maintained at a difference of about 4 to 8 degrees Celsius. For the tropical or subtropical plants that usually grow better at higher temperature, the growth room temperature is maintained at around 28 degree day temperature and 24 degree night temperature. Similarly, for the temperate plants, it is maintained at 25 and 20 degree day and night temperature respectively. Now, temperature also impacts the different stages of the tissue culture. Like in some species, lower temperature usually support the shoot bud differentiation or organogenesis. For example, in tobacco callus, if they are incubated at lower temperature of 18 to 20 degrees Celsius, it improves the organogenesis, whereas the callus perform better at the higher temperature. The temperature within the culture vessel, like the glass tubes or the culture bottles, is usually higher than the surrounding environment in the culture room. This difference in temperature within the culture vessel and the environment can be up to 2 to 3 degrees Celsius and can impact the cultures in the culture vessel. Therefore, to regulate the temperature within the culture vessel, aeration within the vessel is required. We will discuss about this topic in more detail in another lecture. So in summary, we talked about the media composition and the physical parameters that affect the in vitro culturing. We talked in more details about inorganic and organic components, growth regulators, solidifying agents and media pH, and finally light and temperature, how these all different factors they affect the in vitro plant tissue cultures. So with this, it completes the topic of various factors that affect the plant tissue cultures. Thank you.